Okay, so today is our 21st lecture. So we actually finished, now that we reached kind of this point, we actually finished all the primary topics in computational biology that I want to cover for this class. So from here on, will be more like a collection of extra topics or uh, useful tools and knowledge that I added to this class. So for today, we're going to talk about what we can do with bioinformatics tools and database that are online. Before we kind of focus on doing the assignment on our own computers, but now we want to explore what information that we can use to supplement our research that are available online. So today's agenda is simple. We want to consider what are out there kind of on the internet, what types of tools and database that we can explore and look at a lot of them, And that's because kind of many of your research questions, at least kind of part of your research question, some of it may already be found in online databases. Like if you are curious about which, whether you can use certain drug to target a certain gene, like you can either do literature review or you can go to the right database to see the collection of drugs and genes. So let's go to some kind of example of research question that can benefit from online resources. So let's say you have a group of patients with certain disease and you identify some mutation. Usually you want to understand what the mutation does, like in terms of what is impact inside the cell, like does it will it be the likely cause of the disease, like will it be pathogenic, and how rare it is, and how often you can find it in the general population. So this information can help you develop your project, whether you want to focus on certain mutation or other mutation. Let's say you already have done some transcriptomics analysis and you already found differential express gene. In some of our assignment, we try to understand what biological function, what pathways are enriched inside the differential express gene, so that they might be the one that explain the disease, or they might be the one that explain the effect of the treatment. So we already know kind of how to do this, and we can do this with several kind of online tools. We might want to know if other people already study the same gene. Like we want to know if someone already perturbed these genes, maybe with certain compound, maybe with certain drug, or maybe people found that these genes are implicated in a different disease. So those information can help you formulate the biological idea about what this gene does. And you might want to know if these genes can share transcription factors, so they are potentially co-regulated together. And in terms of structure, like we just finished the courses about the structure. So now that we know the structure of the protein, the structure of the RNA, structure of the drug, we can model how these compound and how this protein can be bound together. So we can make a lot of hypotheses about their target and protein-protein binding based on the 3D structure data. And let's say you might kind of identify certain subtype, where you might have a cohort of cancer patients, and you found 3 different subtypes in your patient. And then the next question would be, can you validate this kind of subtype in the broader population in a different country? So instead of doing the actual research yourself, like you can look for online database with maybe gene expression data or mutation profiles for cancer patients in a different country, and then you can confirm if you can find the same biomarkers, you can find the same subtype in those populations. So those evidence will allow you to be more confident about the subtype that you identify in your own analysis. Let's skip some of these examples. So in another cases, if you are more of a computational person, you might want to explore existing datasets, like people that perform large omics cohort, and then you can come into those datasets with a different idea, because not every paper they are not able to analyze everything about that data. There will be some certain factors, like some cell type, maybe some pathways that they don't focus on. not because those cell types or pathways are not important, but because each paper can only accommodate only a few kind of aspects of the analysis. They cannot describe every cell, they cannot describe every function. So there's always space for you to go in to the existing data set, ask a new question, and then get a new findings, get a new knowledge from it. Or sometimes you might come up with new techniques, like a different paper publish a new bioinformatics tools, and you want to apply it to existing data set. That also allow you to identify maybe new genes that the previous techniques couldn't detect, but the new algorithms, like the new AI tools, might be able to find something new. So these are strategies that you can apply to public data. So usually we think about when we combine data from multiple places, like multiple cohorts, as a nightmare for statistics, because you have a lot more batch effect, you have a lot more confounding factors. But if you are able to overcome that, like you are able to apply the right statistical tools to integrate the data, then you have the opportunity, because any pattern that you identify that are true across multiple cohorts, the pattern that can generalize across patients from different countries, like patients with data collected from different omics platforms. If you identify any pattern that can generalize beyond those batches, those are more likely to be real in terms of biology. So even though it's more difficult to analyze, but you have a chance to find new knowledge by combining data sets. So to give you more details on some example, so let's say you identify a certain mutation in the patient. The first thing you can do is you can align your sequencing data against what other people reported in their patients. So to understand if your mutation, is it rare or is it common? That's the first step. You want to know the frequency of the mutation that you found. The next, you might want to understand if the mutation, if it's in a certain protein, will it disrupt the function of that protein? So for the first estimate, you might want to know if that protein interact with other proteins. If this protein interact with many other proteins, then maybe a mutation on this can disrupt how the protein works together. So that can allow you to make hypotheses about what pathways might be affected by this mutation. And once we know the 3D structure of the protein with the mutation, we can ask a more detailed question. Where is the mutation on the 3D structure? And sometimes you can find the 3D structure of proteins that are in complex or in interaction with other compounds, with other proteins. So you can even ask if the mutation will affect the binding between these two proteins. But in this case, the mutation is on the other side of this protein. So the protein of interest are the ILT2 with the blue and the red domains. And the mutation G137 residues is on the outside of the structure, opposite from where this protein binds to the HLA. So in this case, we don't expect this mutation to disrupt the interaction between HLA and this protein. So knowing the 3D structure and the position of the mutation allow you to make new hypotheses, like more detailed hypotheses, or rule out some hypotheses. And nowadays that we have AI like AlphaFold and homology modeling techniques, we can even model the new structure for this protein by introducing the mutation at G137 from G to E, and we can estimate how this mutation will change the overall structure of the protein. So this provides us with even more details than just kind of speculating the effect based on the position. We can actually visualize and model the actual change in the structure using modern prediction tools. Mm-hmm.
Sorry to interrupt you. I want to ask about.
Okay, so nowadays, if you want to understand the effect of a mutation, there are actually new research into the AI model that try to understand the impact of a mutation. So one of them is called alpha missense. So I encourage you to kind of go and look at this paper. So they try to expand the knowledge from AlphaFold. So last time we learned that AlphaFold is an AI that from amino acid sequence, it can predict the 3D structure. So this also means that if you have a wild type amino acid sequence and a mutant amino acid sequence, AlphaFold in theory should be able to predict the 3D structure from both isoforms and also able to compare the structural difference between the two isoforms. So people who develop AlphaFold expand their techniques to try to understand the effect of missense mutation. So missense mutation means that you have a mutation in the coding region that changes the amino acid. So in this case, they will make their model. Basically, underneath, it will predict the 3D structure of both the wild type and the missense variant. And then the change in the 3D structure, combining with other knowledge from coevolution that we mentioned last time, will use to score if the missense mutation is expected to change the protein by a lot or by only a little. So unfortunately, these models, they don't yet release them as a prediction tools. But if you are studying human, and you want to understand the effect of missense mutation on human gene, the AlphaFold structure database also provide the pre-computed, predicted effect of the mutation. So even though you cannot run the tools yourself, if you are studying a known gene in human, you can get the alpha missense prediction from that circle. The other tools that Google released is called Alpha Genome. So Alpha Genome is trained to understand the DNA sequence, not just the amino acid. So while alpha missense focus on missense mutation, so the mutation must be within the coding region. It must be a missense. It cannot be a nonsense mutation or other things. But Alpha Genome, they want to try to generalize the technique so that if you put in any mutation at all onto the human genome, they will try to predict what would be the effect of that mutation. For example, if you try to predict whether that mutation will change the epigenetic profile of the DNA, whether that mutation will change the gene expression pattern in certain cell type, certain tissue type. So this Alpha Genome, they release it as an API on Google Cloud. So if you kind of go read the paper, get the link, you can use Google Mail, Gmail to sign up for the API, and you can kind of use it to predict the effect of your mutation. But these two models are still being evaluated by clinicians, whether they can capture the pathogenicity of the mutation. And we should know shortly whether this AI can be used to supplement clinical application or interpretation of DNA mutation. Okay. For the second example, so this one is a case where you can combine data, transcriptomic data from 6 different hospitals, 6 different research teams in order to get a new biological knowledge. So the situation back then, and more than 10 years ago, was that if you have a colorectal cancer and you go to different hospitals, you might get classified into different subtypes. And this subtype will dictate how the clinician select the treatment for you. So if you have a subtype number 1, you might get radiotherapy. If you get subtype number 2, you might get a certain drug that can treat your cancer. So knowing the subtype is important because it directs the patients to the right treatment. But the problem back then was that when everyone first studied about colorectal cancer, different laboratory, different hospital make different decisions. So if you go to hospital A in yellow, you will get classified into one of the 5 subtypes. But if you, with the same disease, if you go to hospital number 2, hospital B, then you get classified not as one of the 5 subtypes, but with 6 subtypes. And if you go to hospital number 3, you will get classified with only 3 subtypes. So it's clearly that they are making a different decision, even though from the perspective of the patient, you have the same mutation, you have the same transcriptomic profile, but you got classified with different terms, and you might be directed to different treatment depending on where you get your care, and that's bad. We want if we assume that colorectal cancer have the same kind of underlying molecular mechanisms, right? Wherever you go in the world, right, you should get the same decision from the doctors. So in this study, they combine transcriptomic profile of these patients from 6 different labs. And the question that they're asking is, can we identify if these 5 subtypes that group A use, these 6 subtypes that group B use, these 3 subtypes that group C use, these 3 subtypes that group D use, do they share anything in common? Can we map group A1, right, to B2? Can we match group A1 to C2, for example? So what they did in this study is they looked at the transcriptomic profile of each patient. Right? You select one patient. So there's only one transcriptomic profile. And they asked, with this transcriptomic profile, if they follow the guideline from hospital number 1 in yellow, how would this patient be called? This patient might be called mesenchymal. So this patient, if they go to hospital number 1, then he or she will be called mesenchymal. If you take the same transcriptomic profile for the same patient, go to hospital number 2, that patient is called cancer sepsis. If you take the same transcriptomic profile and go to hospital number 3, that patient is called C-type. So by doing this, you can identify that, wait, if I look at the same patient, then hospital number 1 call that patient as mesenchymal, hospital number 2 call them as cancer stem cell, hospital number 3 call as C-type, hospital number 4 call as CCS3, hospital number 5 call as stem cell, hospital number 6 sometimes called 1.1, sometimes called 1.3. So by doing this kind of analysis, by aggregating the data together, the authors identify 4 kind of major groups of patients that whichever hospital these patients go through, they will be classified as these specific subtypes. So in essence, there are 4 kind of major subtypes in the world, and some hospitals classify this subtype into 2 groups, mesenchymal and mixed, or 1.1 and 1.3. But some hospitals group them together into just a single subtype. And that kind of explains the difference in number of subtypes across the hospital, like from 5 to 6 to 3, 5, 5. So by combining data from multiple studies together, without doing anything extra, there is no extra experimental, there is no extra data collection. We can construct the consensus subtyping system that can be used for the entire world. And nowadays, if you have colorectal cancer, and you go to the hospital and you get your transcriptomic profiles from the cancer tissue, you're going to be classified into one of these 4 subtypes. And each of them will dictate which treatment you will get. So this is a good example of how, if you have the right question, you go collect the data that's already been published, you can still make a very big, very important discovery by analyzing them together with your ideas. So hopefully, these 2 examples will give you the motivation of why this class, in particular, focus only on online data, public data that we can collect. So the first type of resources that I want you to be familiar with are called knowledge base. So these are databases that contain knowledge. They call it knowledge base because it contains knowledge. It doesn't give you the raw data. It doesn't give you any prediction, but this will give you the knowledge. For example, if you go to UniProt, it's a resource for proteins. So when you go to UniProt, this might be small, but when you select a protein, they will tell you about the function for that protein. They will tell you about the localization of that protein inside the cell. They will tell you about the post-translational modification that occur on that protein. They will tell you about the expression level for that protein in different tissues. They will tell you about the 3D structure of that protein and more. So basically, you go to one knowledge base. You will get to learn everything about a protein, everything about a gene, or everything about a drug, for example. And these are usually supported by the government, usually in the US, in the EU, or in Japan. So usually they are fully filled for the resources. And nowadays, I think China is also building their own this type of knowledge base as well. So for example, I think you're familiar with NCBI and GenBank from the US, where you can go and get a lot of information about genomes, gene, protein, everything. The equivalent of NCBI in Europe is Ensemble. You probably have seen this before. The other one we will explore in the few slides. So one thing that we have seen in several lectures before, and will be very important when we navigate the online knowledge bases, are the knowledge of these accession ID. As a biologist, you are probably familiar with gene symbol. LGR5 is a gene symbol that you can recognize. But if you go to online database, because gene symbol can change over time, depending on how much we know about the gene, and depending on if new genes are discovered. So this online database will have a different accession system for referring to a specific gene, a specific protein, a specific genome, a specific species. And hopefully by now, you are already familiar with some of the system. For example, if you see MM underscore some number, these are the format for the reference sequence from NCBI, from GenBank. If you see ENSG followed by some number, this is the gene accession for Ensemble. So ENS here is stand for Ensemble and G for gene. And if you go to UniProt, the ID for the protein are usually one or two alphabets, usually O, P, Q, and then followed by 5 numbers. But there are some variation to this. If you see this kind of format that starts with certain character, specific character, followed by numbers, these are usually UniProt accession. But some database can be confusing, like human genome database, NCBI, and omics. Some database only use numbers. So sometimes it's difficult to tell whether when you see a number, does it stand for human gene symbol number or does it stand for NCBI gene number. Even though these are both LGR5. They can be 4504 or can be 8549, depending on which database. So it can be confusing sometimes. Okay. So let's go over some example of the online resources. So let's say you are interested in epigenetics data. You want to know on the DNA, on the chromosomes, which part is euchromatin, which part is generally heterochromatin. Where is the transcription factor binding site? Where is the histone methylation profiles on the chromosomes? If you go to ENCODE, they will show you this kind of track data. So the first track here is the position of the genome. So these are base pair number. And then depending on which epigenetic information you want to look at, they will show you the position, maybe the methylation profiles, maybe the accessibility of the chromatin, maybe the binding site for certain protein. So ENCODE basically contains the processed data from ChIP-seq, from ATAC-seq, and they visualize for you on their website. So you can explore, for example, if you have a certain gene, and you want to know if the 5' region of the gene, does it contain the transcription factor binding site, you can zoom in on that genomic region on ENCODE, and then you can check to see if there is a transcription factor binding site in that region. So ENCODE is all about epigenetics, everything you want to know, even the link to the raw data, raw sequencing data, you can find on the ENCODE website. In another situation, if you want to make sure that the gene that you are interested in Like sometimes you want to explore an omics database and you search for your gene, like LGR5. Sometimes you don't find in the dataset. And that can be because maybe the sequencing data doesn't contain LGR5. Like it might be, the expression might be too low for the RNA-seq to detect. But sometimes it's because each gene can be called by different names. And these are called aliases or synonyms for the gene. So for example, LGR5, in some datasets, they might be called GPR49. In some datasets, might be called GPR67. Some datasets might be called FEX. And the reason is because when we use gene symbol like LGR5, the reason why we are familiar with this system is because gene symbols are usually an acronym for the function or for the knowledge that we have about the gene. So LGR5 is called LGR5 because it's a leucine with an L, like rich, repeat, containing G protein couple receptor. So there is L, G, P, L, G, and R for receptors. So if when the gene is first discovered, we didn't know anything about the function of the gene. So it could get a different name because at the time that the gene was discovered, it was first defined. That's the best knowledge that we have about the gene. But as time went on, as people know more and more about the gene, the name can change. So sometimes when you analyze omics data, you couldn't find LGR5, but when you change to a different name, you can find them. So GeneCard is a website that I usually use to check for the gene aliases. So this website will tell you all the other possible names for a gene, so that you can make sure that the omics dataset that you are looking at really doesn't contain the gene that you want, and not because the platform or the analysis was performed using a different gene name. In another situation, you might have a DNA sequence of interest. You might get it from a chip-seq, and you want to know if in this chip-seq region is there a binding site for certain transcription factor. In order to know, like if your sequence contains a transcription factor binding site, you need to know the binding motif. So JASPA is a database of DNA binding motif for every protein. So if you go to this website and you search for a transcription factor, in this example is a SMAP protein, you can identify this sequence, and you can use this to search in your nucleotide sequence to determine if the SMAP protein can bind to that region. Okay. In another example, you might need to create a table of all genes, all proteins, along with maybe annotation, like the functional annotation. When you use bioinformatics tools, some bioinformatics tools will ask you for this type of data. They want to know all the genes that you want to analyze, and they want to know the annotation of the gene that you want to analyze. So in order to create this type of table, you can go to the Biomart service on Ensemble website. So Biomart, as the name can imply, is a biology, is a market, a mart for biology knowledge. So on Biomart, you can select, first you can select the species of interest. In this example, I select a human. And once you select the species, you can fill in your basket as you go shopping. So in this case, the attributes are the items that you want to shop, the things that you want to export from the database. For example, you can export the gene ID, you can export the transcript ID, you can export the protein data bank ID, which corresponds to 3D structure of the protein. You can also make a filter, like you want genes only on chromosome 1, chromosome 4, chromosome 5. So Biomart will allow you to create this type of table that contains all the genes, all the proteins that you are interested in, along with any annotation that you want. And you can feed these into other bioinformatics tools that ask you for this type of table. So Biomart is useful for supplying annotation data to different bioinformatics tools. In another situation, let's say you want to learn about a certain disease, right? Or maybe you want to understand the genes that are affected by certain drugs. M6DB is a database of gene sets. So basically, they will curate genes that have the same function, maybe genes that are mutated in the same disease, maybe gene whose expression level change in the same disease, or maybe by the same drug, or maybe genes that are published in a paper. Like you might be interested in certain paper that study that identify the gene marker, but most of the time, many publications doesn't give you the Excel table of those genes. So M6DB has curated gene sets that are published in some major study so that you can download them as a table. So it's more convenient for you to find the gene set, the gene list, rather than going to the supplementary data. The limitation is M6DB is only curated data for human and mouse. So if you are studying other organisms, you still have to go to the individual papers. But if you are analyzing human and mouse, you can try to find the gene set that correspond to your treatment, your disease, your function on this website. Okay. The last example for the knowledge base would be this Open Target platform. So Open Target is a drug-centric database. So before the example that we saw before are usually based on genes and proteins. But nowadays, now that we know more about drugs, we have more predictions about the drug function, we have more clinical trials about the drug's effects. So, and we also know more about the genetic variants that can interfere with the function of the drug through pharmacogenomics study. So this Open Targets database, you can start with a drug, a compound of interest, and they will take you to a lot of information about the drug, like the function, the chemical formula, the clinical trial that involve these drugs, and the mutation in the human genome that can interfere with the activity of the drug. So you can get more information before you proceed to do experiment with this drug. So these are the first group of example of online website and online database that will give you a bunch of information about genes, about proteins, about drugs. So the next series of online resources that I want to introduce are where you can find the raw data. Like when people make omics measurement, when they release their data, you can get them from these repositories. So the first one, Sequence Grid Archive, is a repository for high throughput sequencing data. So anyone who perform DNA sequencing, RNA sequencing, ChIP-seq, ATAC-seq, anything that has to do with high throughput sequencing, most of the journal will encourage or even force the authors to make their data public, available on Sequence Grid Archive. So Sequence Grid Archive is hosted by NCBI on the US server. The European equivalent is called European Nucleotide Archive, or ENA, and the Japanese equivalent is DDBJ. So you should know all three, because sometimes, depending on where the researcher is located, he or she might opt to upload to one of the three servers. And even though these three servers, they want to make sure that every data are synchronized across the three servers. There are cases that I found that certain data can only be found on the European server and not on the US, not on Japan. And some data can be found on the Japanese server, but not on the European and on the US. So to give you the best coverage, if you want to identify the sequencing data, you should go to all three to check which one that the authors deposited their data to. So on these servers, on this repository, you can start from a publication and you want to get the data for that publication before you go to the repository. But you can also go to this repository directly and search, for example, by the disease name or by the drug name. and you want to know if there is any sequencing data that were performed related to those disease or related to those drugs. Because in this repository, they will also contain the metadata, the study that generated the data, and other information about the samples, the condition of the samples. So you can either start with specific papers to get their data, or you can go directly to this website and search for the disease, search for the condition that you are interested in. The same idea applies to these three databases for mass spectrometry data, Prime, Massive, and Proteome Exchange. So these are the sequence-read archive equivalent for mass spectrometry data. So people who publish proteomics results are encouraged to put their raw mass spectrometry data onto this platform. So you can either start with a paper and go to get the data from this platform, or you can go directly to this platform and search using the disease, the drug names, and the protein name that you want to see if there is any proteomics data for those conditions. And a special case for proteomics data is called PEP query. So PEP query is a specific database for proteomics that are related to cancer. So they only curate mass spectrometry data from cancer tissues. But because they focus only on cancer tissues, so not only you can search for the data, you can also search for a specific peptide. So basically, if you suspect that your a certain type of cancer might be expressing a certain novel peptide, maybe some new antigen, you want to know if they are expressed in different type of cancers or not. You can put in your peptide or protein sequence onto PEP query, and they will search that peptide protein against their database of mass spectrometry data and give back to you if your novel peptide can be detected in any of these cohorts. So usually a mass spectrometry server doesn't allow you to search for your peptide because that's computationally consuming. But for PEP query, because they focus on only a small subset of cancer-related proteomics, they implement the function for you to search for specific peptide protein. And even though I say that it's a small subset of proteomics, they still have 1,000 million, 1 trillion, 1 trillion, 1 billion, 1 billion mass spectra on that server. So it's not small. So they implement like a fast method for searching that allow you to query your peptide. Okay. But if you want the raw data for cancer, in the US, there is the genomics data commons that collect major cancer study, usually the one that are funded by the NIH. If you get funded, you are obligated to make your data available onto the genomic data common platform. So this is like a federal platform that are available to the public. You can find exome sequencing data, RNA sequencing data, and DNA methylation data on this platform for many types of cancers. And if you are lucky, in some cohort, in some study, they also provide the clinical data and the demographic data. So you can even analyze the survival profiles of the patient from different cancer with different transcriptomic profile, for example. And in some study, they will link the omics data to histopathological image and radiological image. So you can even perform something like a association between mutation profiles and the image of the cells. Sometimes you can tell that the morphology of the cancer cells change because of the mutation or the change in transcriptomics. So nowadays, many people are trying to understand change at the molecular level that usually require omics analysis. But instead, they want to be able to tell the omics result, like the gene marker, the gene expression level, or the mutation profile by just looking at the image, like looking at the histopathological image or radiological image. And you can tell if the patient has a certain type of cancer. So that will reduce the cost of omics. So I mentioned this because in the next lecture, we're going to talk about techniques, computational techniques that can analyze image data. So you can look forward to the next lecture. Okay. For the next resource, if you want to obtain 3D structure for the protein that are already known through experiments, not the one that are predicted, you can go to the protein data bank or PDB. That's the format for the protein 3D structure. You can download the structure of proteins, RNA, and maybe protein in complex with other proteins, with other ligands. If the structure has been experimentally solved with X-ray crystallography, for example, you can download them from this website. Okay. If you want transcriptomic result, the gene expression data specifically, and you don't want the raw sequencing data, hopefully you want the table of gene expression, because that's easier to use. You can go to Gene Expression Omnibus and Array Express. So Gene Expression Omnibus is on NCBI in the US. Array Express is in the EU. But both of them have the same function. For study that produce usually gene expression data, but also if you produce ATAC data or ChIP-seq data, and you already process the data, you can release that table, like the gene expression as read count, or microarray as intensity, or ChIP-seq as the height of the peak. You can release this processed data to these two repositories. And similar to what I mentioned before, even if you don't have a target paper that you want to get the data from, you can go to this platform, search for certain disease, certain condition, and they will show you if there is any processed omics data that correspond to these disease or conditions or treatment. So you can collect a large cohort of expression data or other data by going to this. The last example of repository that I will mention are the ones that are not specific to biology, but it turns out contains a lot of biological data. So Google Dataset and Figshare. So Google Dataset is like a search engine, like a Google search, but it's a search that are specific for dataset. So I forgot the exact detail of how they figure out which website or which file is a dataset, but basically they optimize their search to give you datasets that have your keywords. So you can basically search the entire internet to find some data. Figshare, if you are not familiar, so Figshare is a place, it's a repository, but it is not specific to certain type of data. Usually the repository that we saw before will be specific to certain type of omics data, certain type of disease, for example. But for Figshare, is a generalized platform. Basically, you are given, let's say, 20 gigabytes. You can upload any files you want. You can create any data set you want. They will ask you to fill the metadata so that you can describe the data collection process. You can describe the conditions of the samples. And many journals, when they require you to make your data public, if it is standard omics data, you should go to the standard repository. But if the data is a special type of data, like you might have some images, you might have some process tables that you want to release, there's no repository for those type of data. So you can upload your data to Figshare, and they will give you the DOI number that you can refer to. So this way you can make your data official for type of data that don't have the standard omics database. Okay. So that's the second type of resource where you can go get the actual data. The next type of resource is specific for human, because we are in medical school. So if you study omics data from human, especially gene expression, you are in luck. There are many resources that you can use. For example, the GTEx database. gene tissue expression database. It's a database that will show you the tissue specificity for a gene or for transcript. So basically, they perform transcriptomic analysis of many human tissues. And based on these results, if you have a certain gene of interest, you can confirm whether that gene, whether it's expressed in the tissue that you are interested in or not. Or if you have a certain markers for the cell type or for the tissue type, you can confirm with G-text whether your genes is a good marker for selecting different tissues or different cells. So if you are interested in human primate data, you can look here. If you want to explore single cell, like we learn about single cells data. If you want to get single cell data for specific human tissues, maybe normal tissues, maybe disease tissue, and you can go to the human cell atlas. You can search for the organ, or you can search for the disease. They will point you to the papers that publish the data, and they will give you the actual single cell sequencing data in the form that you can download and use in standard bioinformatics pipeline. But if your goal is to just visualize the cell types, like how many cell types are there in the sample, Human Cell Atlas itself doesn't give you the visualization tools. So you have to download the data and run some tools on your computers. But nowadays, if you are familiar with the UCSC, in the past they made the UCSC genome browser that you can visualize the genomes for different organisms. Nowadays, they also create this UCSC cell browser that allow you to visualize public single cell data. So the dataset that you can find on Human Cell Atlas, you can download them. But if you want to visualize them, you can go to the UCSC cell browser, and you will be able to search based on the organ, based on the disease, based on the organisms. And you will be able to view the dimensionally reduced version of the transcriptomic data. And in here, if you click on different cell type, they will even show you the markers, the genes that are upregulated in this cell type. And you can visualize this by changing from cell type to the expression level of specific genes. So if you go explore these tools, you will find that you can see really large single cell dataset without having to run the bioinformatics tools yourself. The other resource that are very useful is the Human Protein Atlas. So Human Protein Atlas try to give you the complete information about protein, so similar to UniProt. But they focus on human protein. And because they focus on human protein, they can give you a more visualization than UniProt. So if you go to UniProt, you learn about the protein, but usually there will be text that you have to read. But for Human Protein Atlas, they will show you, I don't have all the examples here, but you can even look at the tissue image. You can look at even protein in the blood sample. And you can also compare the expression of protein across different diseases. So you can try to identify if certain protein is a good biomarker for certain type of disease. So this is like a very focused resource for human. Okay. So the next set of resources that I want to show will be the one that allow you to see the interaction. So I think 3 or 4 lectures ago, we learned about networks, like how we draw relationship between genes and protein, and how we can learn from those visualizations. So these tools will allow you to go get the data or visualize the data to see the interaction between multiple proteins that you are interested in. So the first one is called BioGrid. So if you go to BioGrid, you can search for a certain protein, and then they will give you all the other proteins that are known to interact, are known to bind to the target protein. And in BioGrid, they will focus on experimental data. So you will have the information about what kind of experiment that suggests the interaction between the protein. Is it the yeast-2 hybrid assay? Is it the affinity capture mass spectrometry? Is it cross-linking? And these are the papers that you can check to make sure that you can trust the result, whether these two proteins are actually interacting. So BioGrid is good if you want the interaction. You want to download the interaction. But if you want to visualize, like you want to view the data, there are two additional tools that you can use. So if you go to Wikipathway or CAPE, these are also database of protein interaction, but they are organized in terms of biological process or biological pathways. And this diagram is compatible with bioinformatics tools, so you can download the pathway diagram and you can add your gene expression data. For example, you want to view, like in this pathway, if it's upregulated or downregulated as a whole. You can visualize the differential expression from your transcriptomics or from your proteomics onto each individual genes, so or protein. So you can see that the protein that are in red, these are upregulated in your treatment conditions. The protein that are in blue are downregulated. So you can kind of evaluate whether the differential expression result, does it agree with the activity of the pathway? If these are activating each other, should they be in the same direction of regulation? If some of the protein are replacing the other protein, their expression level should be in the opposite direction, for example. So that's one possibility. You can download the pathway from this data set and then visualize them together with differential expression result to see if the pathway is up or down regulated. The second option, the second tools that are actually in the assignment is called string. So string is a visualization for protein-protein interaction. But in contrast to BioGrid, so BioGrid will give you data that are confirmed by experiment. Have to be very rapid confirmation for the interaction. But because there are many possibilities of protein, not all the interaction can be confirmed by experiment. So in string, one of the additional benefits that they provide for you is they also try to predict interaction that you might be interested in. So in addition to known interaction validated by experiment, they will show you predictions. So for example, if you have two proteins that when you read papers, like if you read biological papers, you see these two proteins being mentioned together, right? So they appear in the same paper. Very often, what would be the possibility? Like, if a paper mentions two proteins, it might be because these two proteins are affected in the same disease that the paper is about. Or these two proteins might be affected by the same treatment that the paper is talking about. Or these two proteins might appear in the same function that the paper is talking about. So the fact that if you go through biological papers and you find two proteins that are mentioned together very often, that's a good evidence that maybe these two proteins work together somehow in some context. So in string, they will draw the line between these two proteins as well. Proteins that co-occur together in the same paper. Another example, if you have groups of proteins that maybe in bacteria, none of them exist. So in bacteria, the bacteria don't have these proteins. But when you go to higher and higher organisms, you start to see these proteins being expressed together. So that's an evolutionary evidence that maybe these proteins perform some function together, because in bacteria that doesn't need this function, there is no protein. In higher organisms that need this function, you see all the protein. So the core evolution pattern like this can also suggest that the protein are related somehow. So this relationship in terms of evolution will also be drawn in the string. So basically string will try to give you more hypotheses about protein-protein interaction that you can use to understand if two proteins are related together or not. Okay, let's move on to the next series of online resources. So now we will go to the tools, like bioinformatics tools that you can run online, or prediction tools that you can run online. So for those of you who study microRNA, like sometimes you want to predict, like if you have a gene, you want to downregulate this gene with a microRNA. So you want to know, like given a gene, what are the microRNA that can bind to the 3' UTR for those genes. Or maybe you already done some microRNA sequencing and you found that certain microRNA is upregulated in the after treatment. So you want to know like which gene are affected by these microRNA that are upregulated. So target scan, MIR-based and MIR-DB. These are online prediction tools that will either, if you put in a gene name, it will give you back the microRNA that can potentially bind and regulate this gene. If you start with a microRNA, they will give you back the list of genes that might be regulated by the microRNA that you put in. So this is a resource for predicting genes and microRNA interaction. If you want to identify DNA binding motif, so in the earlier slides, we mentioned that we can go to Jasper and then download the binding motif and then search the data ourselves. But why do that? Right? When you can go to the meme suite, it's a collection of tools related to DNA motif analysis. You can put in your DNA sequence and they will use the database in Jasper that has all the known DNA binding motif. They will use those knowledge to search your DNA sequence and tell you back what are the transcription factors that can bind to your sequence. And they can even calculate the enrichment for you. If you put in multiple sequence, what are the transcription factors that are enriched among your input sequence? So you can run these tools online, or you can download them and run on your computer as well. Okay. The next tool is Swiss model. So if you want to predict protein 3D structure, before using AI like AlphaFold, if the protein that you are interested in have a similar protein with known structure that has been analyzed in the past, Swiss model can perform homology modeling. So you use the known structure as a template and then fit in your new amino acid sequence onto the structure. I recommend this one before AI because with Swiss model, with this kind of homology modeling prediction, they not only give you the prediction, they give you the quality of the prediction. For example, I think I forgot if I mentioned this in the structure lecture or not, but the Ramachandran plot, you can explore whether this structure model is the angle between amino acid residues. Does it follow the expected angle of what people know about the bonding of peptides or not? So this is like a quality estimate for the correctness of the structure. And there are also other scores that they will calculate for you, whether the structure looks natural or does it violate certain laws of chemistry or physics that people know about protein. So Swiss model not only give you the predicted structure, they give you a more detailed quality check. So I recommend using this as well. But if you found that in Swiss model, because it requires a known structure as a template, sometimes you have a new protein sequence, maybe from a new species, maybe a protein that you design in the lab. They don't have a known template, and you need to use AlphaFold. So nowadays, AlphaFold version 3, you can sign up with your Gmail, and you can add your nucleotide sequence or protein sequence. You can even add more than one protein, and they will optimize the 3D structure for you. So AlphaFold will be able to give you the confidence score for each region. In blue means very high confidence. In yellow and in orange means very low confidence. But it's not as detailed as a service model. There is no bond angle analysis. There is no other quality analysis. So I recommend kind of trying both and compare the results. Okay. So with the capability that we can start with amino acid sequence and then we can predict the 3D structure. So nowadays, people try to use this ability to improve the way that we identify similar protein. If we go back to the the sequence alignment lecture, I think that was our second session. We mentioned that if we have a protein and we want to identify similar protein, we can do that with sequence alignment. We can do BLAST-P. We can search for similar protein sequence. And the hope is that similar protein sequence will imply that they have the same function. But because protein function is dictated not based on the sequence, but based on the 3D structure. So it would be a better idea if you have a protein sequence, you predict the 3D structure first, and then you use this 3D structure to search, not for protein with similar sequence, but for protein with similar predicted structure. Okay. So it will be a structure search instead of a sequence search. So FoldSeq is a tool that you can put in your amino acid sequence. Underneath, it will try to predict the 3D structure and compare that 3D structure to known protein structure or to the predict 3D structure for unknown protein and give you back other protein with similar 3D structure. And hopefully that will give you a better result for protein with similar function because the structures are similar. Even though it's predicted by AI, it's theoretically a better way to find protein with similar function. Okay. With the knowledge about protein structure, people tend that to try to predict for each residue on the protein, what would be the function for that residue. So for example, we know that on the protein, there are some domains, maybe DNA binding domains and somatic domains. And those you can annotate based on the sequence. So nowadays we have AI model that looks at your amino acid sequence and it will give you back this annotation that, okay, the residue number 5, the lysine, and residue number 7, number 8, number 11, these are probably linked to small molecule binding. So it can predict what would the function of each region of the protein should be based on just amino acid sequence. So going forward, like from today, you're going to see more and more prediction tools like this that allow you to make sense of entirely new sequence, DNA sequence or amino acid sequence. Okay. The next set of tools are the ones that allow you to perform bioinformatics analysis online. Some of these you are probably have seen in the assignment or in earlier lectures. In gene expression omnibus, if you look at a certain data set on gene expression omnibus, and if the data set is collected based on microarray, not DNA sequence, not RNA sequencing, but microarray. If it's a microarray data, then you can analyze the data online. You don't need to download the data and analyze them with R or Python. On the gene expression omnibus website, if the dataset is correct using microarray, there will be this button called Analyze with GO2R. And if you use this, you can perform differential expression analysis using the R script that are built in on gene expression omnibus website. Similarly, you are familiar with Galaxy already. You can run many bioinformatics tools online, not without using your own computing resources. GenePattern is a similar platform offered by the Broad Institute in the US that allow you to run a different set of bioinformatics tools as well. And on Galaxy, in your assignment, you usually analyze the data yourself by using the specified bioinformatics tools. But you might be curious to know that on Galaxy, there is a workflow tab. If you go to the workflow tab, there will be other bioinformaticians in the world that create their own pipelines. And they want to share that with everyone so that you don't have to know the tools. Like you can just try to read the description. If you want to assemble genomes that comes from PacBio sequencing data, then you can use this workflow. It will ask you for the sequencing data from the PacBio, but it will do all the assembly, all the QC, and will give you back the result at the end. So there are many kind of pre-made workflow that you can use. And some of them you can check the quality if it's already been reviewed by the community or if it's something unknown. So make sure to check this as well because there's no guarantee that the pipeline that you are getting is the one that's correct. But if you see the one that has been reviewed, at least you can trust the community that these are the pipelines that can work. If your use case, if your objective match the description of the workflow. Okay. Some of these you have used WebJSOL before. So WebJSOL is where you can put in a set of genes and you can get back the enriched biological function or biological process. Another tool that I want to introduce is the G profiler. So they also have the functional annotation tools with a different visualization. So depending on what you like, you can try both services. Okay, so that's kind of everything I want to mention today. I want to emphasize that nowadays there are many websites, many tools that you can run analysis or you can get the data, you can find the knowledge online that you don't need to download everything to your computer, anymore. So being familiar with these tools will allow you to get to the answer faster, allow you to refine your hypothesis in your research. Before you invest in experiment, you can check if the genes or the protein or the drugs are they known, like what are known about them online. If the mutation on a protein that might be related to the function that you are expected, you can check those online before you perform any experiment or collect your own data. And we saw from many experiments that many assignments that we can use online tools to run bioinformatics pipeline and do many things that you need to do. So nowadays it's getting easier and easier to analyze bioinformatics data. But the caution is that from Galaxy, you might already found that there are many settings that we didn't look at when we use DEC, like when we do sequence alignment, when we do differential expression. There are many other settings that we can just leave at default. These can cause problem if your analysis is not compatible with the default setting. So one recommendation that I want to make is even if you don't change the parameters, at least record them. Maybe take a screenshot or note them down in your text to make sure that when you run into problem, you can go back and see, that's the result that I got from this tool with the default settings. So someone can help you fine-tune or change the parameters to solve your problem. Okay, so that's everything I have for today.